I want us to look at question 4, and the question reads, the diagram below represent a seedling. And you've seen that seedling, and it has three parts that have not been able to get given in terms of the naming. So the letters are actually given, or the parts are labeled as A, B, and C. And you might be wondering what a seedling is. And of course, you need to get this from a very important part of what you have in this topic called germination. So I would first want to introduce this term called germination, and this refers to the changes that a seed undergoes developing into a seedling, or you can just say the development of a seed into a seedling. So there are two different ways in which you can be able to define this process called germination. Changes that a seed undergoes developing into a seedling, or the development of a seed into a seedling. So that when you actually end up having a seedling as a young plant going to grow to maturity, then you'd be able to expect now even going into details and trying to look at how this process occurs. And of course, there are two main types of germinations. And of course, the first one is called epigynous germination, and the other one is called hypogynous germination. So we have epigynous germination and then hypogynous germination. So I would first be able to just tell you that germination occurs first when a seed ends up having water entering it through a micropyle as an opening. And the process that involves the water entering into a seed as a seed prepares to germinate is referred to as imbibition. You might find this word somewhere, and it means water entering into a seed through a micropyle as an opening to allow room for germination to occur. So the water entering through the micropyle and entering the seed, of course, creates room for what you call germination to obviously occur, unless otherwise, if other conditions are kept constant. So water has entered into this seed. When the water enters the seed, the food in the seed is hydrolyzed. And after the food has been hydrolyzed, the food gets dissolved by the same water, remember? I've said first the water enters the seed through the micropyle as an opening through a process called imbibition. The water has entered the seed, the water hydrolyzes and dissolves the food. Once the water has hydrolyzed and dissolved the food then gets transported by the same water to the growing regions, the plumule and the radicle, as very key areas inside a seed of this plant, or inside the seed of this part of a plant that's going to grow into a mature plant. So the water has to get transported alongside the food, or the water has to move this food from this area where it has been hydrolyzed and dissolved to the growing regions in the seed, which are the plumule and the radicle. The food has reached the plumule and the radicle. What follows? The water provides a medium in which chemical or metabolic reactions are occurring in this seed. The water provides a medium in which chemical or metabolic reactions will occur in this seed to allow room for germination to occur. There are enzymes, of course 3 of them, that allow room for what you call germination to occur as promoters of germination. We have what you call carbohydrase enzyme, and a good example can be the diastase enzyme. We have what you call the protease enzyme. We have what you call the lipase enzyme. Remember, inside a seed, food gets stored into 3 very important forms. Number 1, we have what you call carbohydrates. Number 2, proteins. And number 3, lipids. So the diastase enzyme breaks down the carbohydrates in the seed, for example, in form of starch, into what you call glucose. The protease enzyme breaks down the proteins in the seed into what you call amino acids. And the lipase enzyme breaks down the lipids into fatty acids and glycerol. Look at what the enzymes are doing inside this seed, ensuring that any food which has been dissolved and hydrolyzed by the water and within the water that provides a medium for these reactions to occur gets converted into fatty acids and glycerol, amino acids, and glucose, respectively, in terms of the lipids, proteins, and carbohydrates such as starch. So we have glucose, we have amino acids, and then we have what you call fatty acids and glycerol. These three soluble forms, of course, carbohydrates are insoluble, proteins are insoluble, and lipids are insoluble. So the enzymes convert the insoluble forms of food stored in the seed into soluble forms, which are glucose, amino acids, and fatty acids and glycerol. These soluble forms of the food inside this seed are able to provide a medium for the seed to now germinate. The same thing you're supposed to notice this one. For example, glucose undergoes a process called aerobic respiration, where it gets broken down to produce energy. And of course, it gets broken down in the presence of oxygen. That's why respiration is a chemical process. Glucose is chemically broken down in the presence of oxygen inside the seed to produce energy. The energy facilitates the division of the cells inside the seed to bring about growth. The same enzymes, apart from converting the insoluble forms of the food inside the seed into soluble forms, can be able to actually convert these soluble forms of the food into plant tissues. And I think you've actually been told about tissues. A tissue is a group of cells that can get synchronized to perform a particular function or function. So the glucose, the amino acids, and the fatty acids and glycerol are able to get converted into tissues in a plant by the same enzyme. So enzymes have two major functions. Number one, to break down the insoluble forms of the food into soluble forms such as glucose, amino acids, and glycerol, and of course fatty acids, and as well being able to convert these soluble forms of the food into plant tissues. Of course, you have so many types of tissue like the epidermal tissue that you're aware of. We have what you call the vascular bundle tissue, the conducting tissue. You're actually aware of that one. We have what you call the gut cell that can be able to form what you call the gut cell tissue. We have what you call the pulsate tissue. We have what you call the spongy mesophyll tissue. And of course, all these tissues perform different functions in these plants. So, and then we have actually had a chance of now seeing these products being very useful to us in the seed. And we are told that now the plumule grows into the first foliage leaves in a seed that is germinating. So the plumule glows into what we call leaves. And the glowing actually makes this seedling to be able to germinate and be able to even make food on its own. The radicle actually develops into what we call the primary root. So the radicle actually exits through what we call the micropyle, breaking the seed and keeping it open so that that radicle grows into the soil as the first root called the primary root. The plumule grows into the first foliage leaves. The radicle cross out of the seed through what we call the micropyle and into the soil, breaking open the seed. So what happens first? The radicle grows out of the micropyle, breaking open the seed and growing into the soil as the primary root. What follows? The plumule growing out of the same broken seed to form what you call the first foliage leaves. Germination has occurred. So what brings about the difference between those two germinations? For epigene germination in dicotyledonous plants, dicotyledonous plants such as peas and beans, and hypogene germination in monocotyledonous plants, such as the grasses. Of course, we have actually talked about the sorghum, the millet, the rice, the maize, you see. So this epigene germination is common in dicotyledonous plants, such as plants that can fall under this category of peas and beans. And of course, monocotyledonous plants that can actually involve different types of monocots, like the maize. We have what you call the grass, the rice, the sorghum, the millet, the wheat. So with this type of germination you've actually seen here, this is epigynous germination. Name the type of germination exhibited by the seedling, epigynous germination. Why is it epigynous? Number one, the cotyledons are brought above the ground after the seed has germinated. The cotyledons are brought above the ground, and of course you can be able to see these cotyledons getting brought above the ground. When these cotyledons get brought above the ground, they turn green and leafy. Once they have turned green and leafy, they begin manufacturing food for this seedling before the first foliage leaves can actually appear or grow out of these cotyledons. So, with epigynous germination in dicotyledonous plants, the cotyledons are brought above the ground, they turn green and leafy, they begin manufacturing food. Being green means they contain chlorophyll that can trap sunlight to help this seedling manufacture food before the first foliage leaves can grow out of these cotyledons. So the plumule within the cotyledons can actually grow into what you call the first foliage leaves. You can look at this part called the hypocotyle. The hypocotyle grows out of the soil, elongating. It grows out of the soil curved, pushing this seedling out of the soil, protecting the delicate cotyledons from getting broken by the soil. So the main purpose of this hypocotyle is to actually curve and grow out of the soil, protecting the delicate cotyledons as this seedling pushes through the soil, coming above the soil or coming out of the ground. Look at this, the root hairs. Their main function is for anchorage, holding this seedling firmly in the soil, and of course absorbing water and dissolved mineral salts for this seedling as it still goes on growing. State the functions of the parts labeled A and C. I've told you A are cotyledons. Remember, they are two. That's why this is a very common type of germination in what you call dicotyledonous plants. Di means two. So there are two cotyledons like the ones for beans, and they actually get brought above the ground. They turn green and leafy. They begin manufacturing food for the seedling as it actually prepares to actually develop the first foliage leaves within the cotyledons, resulting from what you call the plumule. I've talked about the C, which is actually a root hair. Remember, the pointing actually points one hair. So you can refer to that as a root hair cell. The main purpose of the root hair cell is to absorb water and dissolved mineral salts for this seedling. And number two, to hold firmly the seedling in the soil. And of course, we've actually done this. Name the structure labeled B. I've told you it's actually called the hypocotyle. Epigynous hypocotyle. So the opposite. E and then H. So you can be able to actually give that as a hypocotyle. Very good. Now, they have actually talked about the conditions which are actually necessary for germination. We have what you call external conditions that can promote germination and internal conditions. What are these external conditions? We have what you call temperature. We have what you call water. Some seeds will need sunlight to break the dormancy, like the lettuce. You will also be able to need, so you have actually talked about temperature, water, sunlight, and here you can be able to talk about hormones. You can be able to talk about enzymes, and you can be able to talk about viability. So the internal conditions that can favor the germination of a seed. Internal conditions to me they are found within the seed. They do not get introduced into the seed from outside. So they are actually referred to as internal factors or conditions. So hormones are inside the seed, enzymes within the seed, and viability can get talked about when you're referring to the internal structure of the seed in terms of the mature embryo. So then temperature has to actually get associated with the environmental temperature, so it becomes an external condition. We have what we call water that gets introduced into the seed through a process called imbibition via the micropyle as an opening. Then the sunlight from outside, of course, as the main source of energy in biology. And you can also talk about oxygen that also gets introduced into the seed through the same opening called micropyle. Oxygen is responsible for enhancing respiration, where the food within this seed gets broken down chemically to produce energy for the seed to germinate, because the cells must divide. for the seed to germinate. And for the cells to divide, for the seed to germinate, there must be energy. If you talk about temperature, we have talked about enzymes within these seeds. The enzymes are converting the insoluble food products into soluble forms like glucose and then amino acids. Enzymes work best under optimum temperature. Enzymes must be activated. Under low temperatures, enzymes become inactive. They can't be able to actually break down the insoluble food substances into soluble forms. So they must be activated with what you call the optimum temperature. So temperature activates enzymes optimally to ensure that they can be able to actually act on the substrates, breaking them down into soluble forms for the purpose of germination. The water one will actually hydrolyze the food. The water dissolves the food. The water transports the food to the growing regions. The water still activates the enzymes. The water provides a medium in which chemical reactions are going to occur within the seed for us to enhance germination. And of course, to ensure that the seed coat is dissolved and softened so that the radicle can emerge as a primary root and the plumule can also emerge as the first foliage leaves. So softening the seed coat can also be a very important aspect for us to mention in terms of water. Some seeds provide dormancy. They do not germinate even if the conditions are very appropriate. So they require some certain light wavelengths like the leucite. So at the end of the day, you might actually need this certain light wavelength for this seed to germinate because light energy acts as a particular source of energy, a triggering factor, a catalyst to catalyze, to activate these seeds to make them be able to germinate and get out of what you call the dormancy period. Hormones such as cytokinins and gibberellins are very important in seed germination. So cytokinins and gibberellins are very important in seed germination. So cytokinins and gibberellins are very important in seed germination. So cytokinins and gibberellins are examples of hormones responsible for promoting seed germination. If there's a low concentration of these hormones in the seeds may not be able to germinate. So they end up actually entering into a stage or a period called seed dormancy. I've talked about enzymes, viability. Only viable seeds are able to germinate. To mean there must be a mature embryo with enough food reserves within this region to ensure that the seed can germinate. A seed whose embryo is not mature or a seed whose food reserves are actually depleted cannot be able to germinate. We shall say the seed is not viable. So only viable seeds can be able to germinate for that matter. So which question have I not answered? What is the importance of optimum temperature in seed germination? I've actually told you to activate the enzymes optimally, speeding up the conversion of the insoluble food products into soluble forms, which can enhance germination. And I think that is what I had in as far as this question is concerned. I've also told you about this diagram. And of course, if you are actually told to talk more about the hypogel germination, as I've actually told you, it is common in monocotyledonous plants like the maize plants. With this type, the cotyledon is not brought above the ground. The cotyledon remains in the soil. The cotyledon is actually able to still remain within the soil. So it does not get brought above the ground. And what happens at the end of the day, if this is the cotyledon of that plant, you'll be able to have what you call a primary root resulting from the radicle with a protective part called the coleorhiza. Very good. So a radicle grows into a primary root. It grows deep into the soil with a protective part called coleorhiza to protect the delicate tip of this primary root resulting from this radicle. We have what you call a coleoptile that grows out of this seed as well, and the coleoptile pushes through the soil, reaching the ground. And just from this coleoptile is what you have as the plumule that grows into what we call the first foliage leaves. Very good. We have what we call the epicotyle between the plumule and the cotyledon. Remember here, the hypocotyle was between the cotyledons and the radicle, for your information. This is what we call the hypocotyle between the cotyledons and the radicle. For hypocotyle, epigel. For hypogel, epicotyle. So you can get the difference. You can get the difference, the epicotyle. There it is. So epicotyle is between the plumule and the cotyledon. So between or within the coleoptile is the plumule that grows into first foliage leaves, you see. And just below here is what you call the epicotyle that pushes through the soil to facilitate the plumule within the coleoptile growing into the first foliage leaf. So I've talked about the radicle growing into what we call the primary root, protected by a structure called the coleorhiza, as that primary root grows into the soil. I've talked about the epicotyle that grows out of the soil, pushing through the soil and carrying with it the coleoptile. So once the epicotyle reaches the ground level and actually emerges out of the ground, the plumule within the coleoptile grows into the first foliage leaves, and this can begin manufacturing food for the plant. So at the end of the day, the radicle is able to grow into the root, and the cotyledon is not brought above the ground. It remains within the soil. So you can see that this is the ground level. So the cotyledon has remained within the soil. That's what it was actually called the hypogene germination in monocotyledonous plants.